Nucleic acids, specifically DNA and RNA, store and carry the codes for building all the organic molecules needed for life. DNA, deoxyribonucleic acid, stores the code for life. RNA, ribonucleic acid, reads the code from the DNA and delivers the instructions to locations within the cells where organic molecules, mostly proteins, are being built. On the cellular level, life is a cycle. DNA gives instructions to RNA on how to build proteins, and the protein enzymes build more DNA and RNA. Carbohydrates and fats provide the energy and much of the structure to keep the DNA-RNA protein cycle going around throughout the cell's life. In bacteria and other prokaryotes, the DNA floats freely within the bacteria cell. In eukaryote cells, like animal or plant cells, the DNA is separated from the rest of the cell within the nucleus of the cell. The figure shows eukaryote cells have a separate nucleus. In prokaryote bacteria, the DNA float freely in the cytoplasm of the cell. The figure shows an animal cell with its nucleus and other major structures labeled. The nucleus has its own double phospholipid layer nuclear wall or nuclear envelope. The nucleus has several major components including the nuclear envelope, nuclear pores, nucleoplasm, chromatin, and the nucleolus. The chromatin is the tightly wound DNA. The nucleolus is a body that contains the rRNA that construct ribosomes. There are three major types of RNA. Ribosomal RNA, rRNA, messenger RNA, mRNA, and transfer RNA, tRNA. The structure of these RNA strands is similar. It is the functions that are different. Our RNA read the DNA for instruction to build ribosomes inside the nucleolus. The ribosomes are transported out of the nucleus through the nuclear pores and take their place in the cytoplasm or on the rough endoplasmic reticulum. The mRNA reads the DNA for instruction on how to create proteins. The mRNA carries their instructions out of the nucleus and move into a ribosome. The shorter tRNA strands picks up the correct amino acids floating in the cell cytoplasm and carry them to the ribosomes. The ribosomes use the mRNA instructions to assemble the amino acids into peptides that will be folded to become working proteins. The complete DNA sequence to make an organism is called its genome. In multicell creatures, like animals and plants, every cell in that organism has a complete genome in every cell, with the exceptions of specialized cells like red blood cells that have lost their nucleus and DNA. The genome is not on a single DNA molecule. The complete genome of the organism is divided up among sections of DNA called chromosomes. In prokaryotes like bacteria, the chromosomes form rings that float freely in the prokaryote cytoplasm. In eukaryotes, plants and animals, the complete genome is on a fixed number of chromosomes for that species. The human DNA sequence is divided up into 23 pairs of chromosomes. Think the genetics company 23andMe. The complete genome of a horse is divided among 32 pairs of chromosomes. A dog has 39 pairs of chromosomes. A corn plant has 10 pairs of chromosomes. Every species divides up its genome or DNA sequence on a different number of chromosome pairs. Chromosomes come in pairs because of sex. We get one chromosome from each parent. The sex gametes, egg and sperm cells, are haploid cells. The gamete chromosomes come in single strands. When egg and sperm merge, their chromosomes pair up. And from that point forward, every cell in the new organism will get the identical copy of the chromosome pairs that were formed by the merging of the egg and sperm. The individual DNA strands of the chromosomes are called chromatids. When the gametes first merge, the DNA from each parent is recopied, so that sections of DNA from each parent is exchanged or remixed, so that the DNA on each new chromatid will have a mix of the DNA from both parents. A rough analogy might be shuffling blue and pink half decks of cards into each other, so that the child deck might have a blue queen and a pink jack and be a complete deck, but with a mix of cards from each parent deck. Chromosome pairs have an important role during cell division. When the body is growing or needs more cells to replace older cells, the healthy cells divide into two cells called daughter cells. Cell division is called mitosis. During prophase of mitosis, the chromosomes pair up as they were paired in the original fertilized egg. At metaphase, tiny bodies called centrosomes position themselves at opposite sides of the cell. The nuclear cell membrane dissolves, and the fibers called the mitotic spindle connect the two opposing centromeres. The chromosome pairs line up along the equator of the mitotic spindle. At anaphase, the mitotic spindle fibers pull the pairs of chromosomes apart into sister chromatids. At telophase and cytokinesis, the outer cell membrane pinches in the middle to form two daughter cells from the original cell. The nuclear membrane reforms to surround the nuclear chromatin. Each chromosome pair has a section of the genome or complete DNA sequence of its organism. If added together, the complete DNA sequence is extremely long. For eukaryote cells, the complete DNA sequence must fit into the nucleus of each cell. The DNA sequence in one human cell is 5 to 6 feet long, or 2 meters long. The DNA sequence must be wrapped tightly so that it all fits inside a cell nucleus only 6 microns across. The fluid in a cell nucleus is called nucleoplasm. The DNA material in the nucleus of cells is called chromatin. The chromatin is made up of strands of DNA wound around proteins called histones. Histones are formed of groups of 8 histone proteins. The group of 8 histone proteins is called an octamer. Each histone octamer looks like the beads of a necklace. So, a length of DNA coiled around separate histone octamers look like a beaded necklace. The histone proteins contain the amino acids lysine and arginine. The lysine and arginine amino acids have a slight positive charge. The DNA strand has a slight negative charge due to the phosphates on its ribophosphate backbone. So, as DNA wraps around the histones, the DNA sticks to the histone octamer by a small electrostatic charge. The figure shows a zoom down from the cell with a nucleus, down to the chromosome inside the nucleus, down to the histone beads inside the chromosome, down to the double helix of the DNA, and finally down to the individual nucleotides that make up the DNA double helix. So, chromatin is DNA, plus the histone proteins the DNA wraps around. The histones compact the DNA so that the long length of DNA can fit inside the nucleus of a cell. The histones and shepherding proteins organize the DNA so that it can be transcribed by enzymes onto RNA. There are 2 types of chromatins, heterochromatin and euchromatin. When the DNA is tightly wound around the histones, it is called heterochromatin. Heterochromatin is the storage mode of chromatin. Heterochromatin is too tightly wound to be transcribed. When a section or gene of the DNA needs to be read, those sections are converted into more loosely wound euchromatin. This loosening is accomplished by acetylation, attaching acetyl groups ch016a to the histone at the section of DNA that needs to be read. When acetylated, the space between the DNA and the histones opens sufficiently for RNA polymerase enzymes to get into the space between the DNA and the histone, and transcribes the DNA code into a new RNA strand. The RNA polymerase constructs the RNA in place as it reads the DNA. When the polymerase reaches a stopping point, usually the end of a gene, the RNA is released to float off. The RNA leaves the nucleus through a nuclear pore and carries its message to a ribosome, where the instructions can use to build more proteins. The heterochromatin could be loosened by other means beside attaching an acetyl group ch3co to the section of DNA. For example, attaching a single methyl group CH3 to the histone will loosen a heterochromatin section into a euchromatin section so that it can be transcribed. On the other hand, attaching 2 or 3 methyl groups CH3 to the histone at a section of euchromatin will cause the DNA to wrap more tightly to its histone, converting it to heterochromatin and making that section of DNA too tightly wrapped to be transcribed into RNA. The sections of DNA that must be transcribed are called genes. Genes are sequences of DNA code for at least one protein, but sometimes two or more proteins. Methylation results in suppression of the gene that is present on that section of DNA. Suppressing genes through methylation, or opening up genes to expression through acetylation, are topics in epigenetics. Epigenetics is the study and the action of suppressing or expressing genes. The time when a gene is expressed or suppressed is as important as the genetic information carried on the DNA. The DNA molecule in every cell carries the information to turn that cell into a heart cell or a skin cell or a brain cell. It is the epigenetics that control which genes on that DNA are expressed or suppressed, and that guide the cell to grow into its particular role in the body. When epigenetics go awry, the result could be a cancer cell that grows and divides out of control. A possible cure for cancer and other genetic abnormalities makes epigenetics an important field of study. And that concludes my introduction to nucleic acid functions. In my next video, I will discuss the building blocks of DNA and RNA, the nucleotides, and how they are assembled from nucleobases, ribose sugar, and phosphate.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.